In an attempt to discover a method for destroying the bacteria that cause pneumonia, Frederick Griffith conducted experiments on 2 strains of the bacterium Streptococcus pneumoniae. One of these strains caused the death of laboratory animals, while the second was more benign and usually harmless. Griffith assumed that the virulence and harmlessness of bacteria depended on whether or not they produced a shell. One of the strains, referred to as S, smooth, produced a smooth shell composed of sugars. The second strain, referred to as R, rough, did not produce such a shell. Griffith conducted a series of experiments on mice in order to find a vaccine. First, he injected mice with bacteria of the S strain. All the mice became ill and died. Then, by subjecting them to high temperatures, Griffith killed bacteria of the S strain and injected them into mice. As he expected, none of the infected mice developed pneumonia. When Griffith injected the mice with live bacteria of the R strain, they did not become ill. However, when he injected mice with dead bacteria of the S strain and live bacteria of the R strain, that is, the benign strain, the mice developed pneumonia and most of them died. The bodies of dead mice that were given both the S and R strains simultaneously contained living bacteria of the S strain, but no bacteria of the R strain. Griffith concluded that bacteria of the R strain had taken the genes responsible for the formation of cells from bacteria of the S strain, had become virulent, and had passed on the ability to produce shells to the next generations. This ability proved to be permanent and inheritable. The phenomenon of passing genes from one bacterium to another is referred to as transformation.